Hi,
how are you? All right. Well, welcome. So, any updates? Anything we want to talk about before we get started?
Still working on my thesis,
Oh, yeah?
a write-up.
Yeah,
Yeah.
that's good. How is it going? Just,
I had to find a validation for my measurements, and so I did, sort of. Basically, I'm having to write up a section, like a chapter in my thesis for validation, because there is no direct measurements that I can get that are exactly the same as what other people have measured. I'm going to call it a fibroblast. These are all fibroblasts. If you pull on them, they're stiffer. So that's that one, and this is that one. That's one of my conclusions.
yeah.
I'll just call them all fibroblasts.
That's what everyone else does, I guess. It's just kind of like the standard cell type.
Yeah. And if you want lung tissue, well, you can't stretch it more than about 10% or it fluidizes, apparently. So,
Okay.
yeah. Fluidize and tension. That's what they did. Anyways, so. And it's really interesting, their curves. They do have sort of a J curve, but they're all over the place. Like they're like this. And that's what I'm getting for all of my data. They start off going every which way.
Huh.
And it's very, very deterministic. Like you can measure it 10 times and you will get the exact same graph.
Oh, wow.
Yeah. Very stable hexagon graph.
Okay. It's interesting.
Yeah, why should a hexagon be stable like that?
Well, yeah, I think there's geometric stability, of course, but yeah, I don't know.
Yeah. Anyway, they are, at least that. I had another setup where I had the microtubules going the other direction so that it wouldn't stand up at all, and I would get all kinds of different results all the time from it.
Huh. Interesting. Yeah, I did see that you sent some papers to Dick and I. I'll have to look through those. I saw the email. I didn't get a chance to look through them. But
Yeah, that's okay. I was writing another chapter for my thesis, and I ran across a few things, so I thought you would like them.
oh, yeah. Yeah,
Yeah.
Well, that's good. Yeah, we'll have to look at those pretty soon. So, well, good luck with the thesis.
Thank you.
Hopefully, we can see some of the data later when you make sense of it more. And
Well, I'll write it up, hand it in. They'll probably want to rearrange it, and when I've handed it in, I can present it here.
okay, that sounds good. Thanks, Susan. Looks like Hussain is here if he wants to give an update.
Oh yeah, I can give an update. Thanks.
Yeah.
So I've been working with Dick on the breast cancer on the RBRCT project. Yep. Yeah, we got published. Really, really happy.
Yeah, I saw that paper.
Yeah, excuse me. Yeah. That's basically it. Yeah.
Did you want to put a link to the paper in the chat so we can see?
Sure. I mean, it's not public. It's not public yet.
Oh, okay.
We have our PDF. We have the paper.
All right. Okay. Yeah, but you're doing pretty well otherwise.
Yep. Yeah. Happy to also contribute to DevoWorm, to OpenWorm as well too.
Yeah.
If I need to, if there's any way to. Yeah. I will just say with the work projects, at times it can be difficult to figure out what really needs to be worked on. You know, sometimes theories have been raised like maybe 4 or 5 years ago, and it's like tough to know. Okay, but what's relevant right now? So, yeah.
Well, I mean, it's kind of hard to say. We're having our annual meeting next month, so maybe in about a month from now.
Okay, cool.
Yeah, and I think like a lot of the things revolve around the containerization. So we have a Docker container.
Yeah.
Some other things like simulations. We've been talking in the group here about, doing things with CompuCell 3D and some of the other multiphysics models. And, there are other things in OpenWorm, and I'm not sure exactly what. I know Porig Gleason is probably the most active person right now.
Well, yeah, I'm also definitely open to contacting individuals who may know of something in dire need of issue or something. I'm also happy to suggest my own changes. Like if I've been directions for the project, there are things you want to take things or research, software, AI, machine learning perspectives too. Obviously I don't just want to like air out random ideas, and I want to make an informed pitch about something that may be relevant. That's really important. But the other thing I'll mention is that I've become a lot more stable with my career at the EdTech startup in California for that educational technology startup. So I feel a lot more confident and comfortable taking on side projects within limits. So once you like feel like stable with that, then making open source contributions to voluntary projects like OpenWorm actually feels a lot easier.
Yeah.
Like other than when I was applying to like 20 jobs a day sometimes, and it's taking time taking time out of my day to go to the gym felt too difficult because I'm like I should be firing off job applications. But the recent project progress both on the Janice-free, the RBRCT project with Dick, and the work I do with the startup with Alter Learning. So for those of you who don't know, I've been working at a startup in California, Alter Learning, and I spent my time between both of these pursuit projects. Both of these recent successes have been really amazing. We've been using Tapas, and I got Tapas working. So Tapas is Tapas is a simulation tool for Monte Carlo. Using the Monte Carlo method for radiotherapy, like radiation therapy simulations. Excuse me, and we looked into this one among many other software, many other programs and things, and Tapas is working now. So now I can be a lot more creative and liberal with the best ways to visualize, the best ways to move forward with Tapas's data, like what sort of simulations are important for us. And I've even contacted a patents lawyer for our publication, and they've gotten back to me. They're happy to do an art search. So that's called a prior art search to test if a prior art search to determine the qualities of our technology from a patent perspective, to see it's relevant, if it's novel, if there's utility with it, and I'm going to look at my options with them there and with them on the Janus technology. We would be patenting electronic steering of X-rays. That would be the patent. So, and if we can patent that and establish a multi-patent IP portfolio, that's probably I would see that as the best thing moving forward. So, yeah.
Yeah. Well, that's good. I mean, and you're welcome to propose your own contributions to either OpenWorm or DevoWorm. I know you had the open and closed worm repository we're working on a while back.
Yeah.
I mean, like a lot of machine learning stuff I think would be good as well. I mean, there are a lot of problems you could kind of approach using those methods. So yeah, I welcome you to, or we can talk about it more, or just kind of come up with your own thing, and kind of just go ahead.
Yeah, just always saying, these products they come and go, but as long as you like have decent follow up every now and then, I find things very productive and practical. Even just like coming back to something like several months later, and to get back on board with things, because sometimes things happen, things fall to the wayside. It happens here and there. I think like even getting to past running took a month or two of me just messing around with it until I got it working. So things like that happen. It's no big deal. It's all right. Um, but yeah. Really happy to keep moving forward. So, yeah.
Yeah, that's great. Thank you. I don't know if Morgan had a update or if he's in the morning schedule.
I was like, well, yeah. And just hoping to get everybody to school on time.
Yeah, that's good. So yeah, actually, given what Hussain was just talking about, we do have a new data set to look at here. So there's a group of people who it's actually let's see. This is a group of people from, I guess, MIT and then some other people. So Ed Boyden is involved in this. And I know that Conrad Cording actually talked to me a little bit about the C. elegans connectome at one point. But anyways, this is a group that is looking at the C. elegans connectome. And, they have this new data set, this new connectome. So, the connectome in C. elegans is well known. We know all of the cells, we know their nomenclature, and we can model the entire connectome. And that's what's one of the things that's appealing about C. elegans is that you have this relatively small connectome. We know the cells, and it produces behaviors that we can recognize as like something that a complex animal would produce. But anyways, um, so this is a data set I think that they've either generated or compiled. And so this is on Hugging Face. So, they have repositories on Hugging Face. And this is C. elegans connectome data. Q. Simeon is the contributor. And the this is from, I guess, a conference, Southeast Con. And the paper was called Scaling Properties for Artificial Neural Network Models of a Small Nervous System. So the figure is here: compiled labeled connectomes of C. elegans from multiple experimental sources, standardized to a common graph tensor organization format. These tensor files are formatted as following with the relevant accessible attributes and methods. So this is where they've taken different connectome models from the literature. We have a hermaphrodite connectome, we have a male connectome, and we even have like a developmental connectome. And usually, most of the data is based on gap junctions. So the idea is that if two cells have a gap junction, they're connected. And so you can draw out this graph. And of course, they're also like synaptic based connectomes as well. But I think this is just basically from different gap junction connectomes with what they call electrical connections. And it's from multiple experimental sources, so they're kind of compiling everything. And then they're transforming this into a graph tensor format. So they're getting this ready for machine learning, basically. So this is where this is an example of the data structure here, graph tensors. There's an edge index, edge attributes, some information about the graph, the number classes, the X and Y coordinates, and some other labeling information there. Some important notes. Number one is the edge index attribute, or edge underscore index attribute, contains directed edge connection information between node pairs. So edge index is like where you get the connectivity information. So if two cells are connected, they have this value. Number two is that the edge attribute, or the edge underscore ATTR attribute, contains gap junction and chemical synapse weightage. So they do include chemical synapses as well as the gap junctions or the electrical gap junctions. So they have both of those in here. And so this is, it contains gap junction and chemical synapse weightage values aligned with edge underscore index directed edges. So they basically have this connectivity information. They have the weights and aligning it with directed edges, so you know kind of in which direction there's a connection and what the weight is. And then the third point here is that the positive attribute or POS attribute contains 3D location information for each neuron. So this is where you have, I guess it's position, not positive, but it's this, basically I guess a three tuple where you have the 3D location. So this is how you might execute this in a Colab notebook. And this is the kind of the code. Graph tensors, you load PyTorch, I guess. You go through this and you sort of define everything and you draw the connectome in graph plot 3D false. So let's see. This is a visualization of said network. So this is how they're visualizing it. They have the labels, they have the categories, they have the connections in red, and you can see the connections, how they're directed, because you have arrows going from one set of neurons to some other neurons out here. And you have you have both types of connections, gap junction, synapse. The red ones are synapse. The blue ones are gap junction. I don't think they're showing gap junction in this image. But the idea is that, and then this kind of circle here is the edge of the worm, I guess. So this is just kind of showing the connectome sort of in place in the anatomy, showing the connections, taking all these data and putting them together. Which is nice because, there are a lot of data sets out there that are kind of, independent. And so it's nice to have this here. So I guess this is kind of their first take at, building this for some sort of machine learning purpose and getting the data into shape for that. Any questions about that or comments? Okay, so that's available. That is on Hugging Face. That's Datasets, Hussain, and then C. elegans connectome data. The other things I want to talk about. So this is actually the Turlier lab, and they're based, I think, in Paris. And they have some interesting stuff on their GitHub, and I wanted to point this out. So the Turlier lab, it's called their sort of username is Virtual Embryo on GitHub. And so they have a lot of different repositories. They have this foam embryo or foambryo repository, which is this work that they did in a number of papers where they were able to infer tensions and pressures from 3D microscopy images of embryos and tissues. So they wrote a paper on this a while back, and I'm trying to remember what the citation was. The citation is here, yeah, where I think it's a bioRxiv paper. Yeah, this is the paper, Embryo Mechanics Cartography, Inference of 3D Force Atlases from Fluorescent Microscopy. So this is Hervé Turlier and colleagues. And, this is the paper here where they do this measurement of forces at the whole embryo scale. So they do these foam simulations. So basically, they want to understand the morphogenesis of tissues and embryos and the role of mechanics in that. And so we can get sequencing, we can use sequencing methods to get spatiotemporal maps of gene expression, but we can't necessarily get a map of cell mechanics in the same way. So that's what they want to do here. They're basically treating the cells in the embryo is a foam-like geometry. So they're using the soft materials approach to modeling the embryo and the cells in the embryo. So they call it the foam-like geometry of cells and embryos. We propose a robust end-to-end computational method to infer spatio-temporal atlases of cellular forces from fluorescence microscopy images of cell membranes. Our method generates precise 3D meshes of cell geometry and successfully predicts relative cell surface tensions and pressures in the tissue. We validate it with 3D foam simulations, study its noise sensitivity, and prove its biological relevance in mouse, Ascidian, and C. elegans embryos. So they actually use a wide variety of embryos, including the Ascidian or the C squirt and the mouse. So we have, a number of different modes of development there. 3D inference allows us to recover mechanical features identified previously, but also predicts new ones, unveiling potential new insights on the spatio-temporal regulation of cell mechanics in early embryos. Our code is freely available and paves the way for unraveling the unknown mechanochemical feedbacks that control embryo and tissue morphogenesis. So if we go back to the repository, we have the README, and if you go here, I guess we have information about their model organisms, and yeah, so they have some images here. This is equilibrium. This is the force viewer. So this is the sort of the software they're using, this polyscope, where they're able to kind of model the cells. And I think maybe it'd be better to go back to the README to see if they have any information about how to run this or what to download. So basically, you build these simulations. Basically, you have to run foam embryo with Python. It's Python code. So you plug it in. You can use, some algorithm to do image segmentation. So you have to segment the images of the embryo you're interested in. You have to create masks. Then you have to basically run the software, run the analysis, and then you can view it in Polyscope, there's a link to it here. So this is a way to visualize this 3D geometry. So it's this software, you basically do the segmentation, use the software to do the analysis, and then you visualize it in Polyscope. So that's basically what you're doing. So, it's pretty easy to install, pip install foam embryo. You install the package, and then you start doing your work. So you're taking things from a segmentation mask, and you're doing the analysis, and then you dump it into this viewer. So this is where we have the API and documentation. So they have actually a pretty good layout of how to use this and showing the examples and everything. So this is nice software here. I just thought it might be interesting, especially for Susan. And they have some other things here as well going on. A lot of things with mechanics and the physics of the embryo. Okay, the third thing I wanted to talk about was this embryo multiscale modeling course. This is something that Morgan pointed out, I think, in the Slack, and he gave a link to it. So this is a multiscale modeling course. This is EMBRIO, EMBRIO, which is an abbreviation of some type. So basically, it's a modeling course where you can go through and follow along and create your own, sort of get a lot of knowledge about multi-scale modeling and apply it to your favorite system. So basically, you go through the introduction. There's this introduction to scientific computing, partial differential equations and biological systems. So a number of examples of that. And then equations of state, going through that, signaling pathway dynamics. So we're going through kinetics, biochemical kinetics, reactor stability and sensitivity, looking at energy balance, and steady state energy, energetics, and then morphogen gradients, kind of going through that. Reaction diffusion in an antibody detection device. That's a project that they're offering here, kind of a sample project to go through and work out on your own. So you kind of get the background up above, and then you start doing this project. There's another project, max toxin manufacturing, which is an antitoxin. Another project on circadian rhythms, where you can model, feedback loops and oscillators, phosphorylation states, and other things like that. Model chemotaxis adaptation, where you have different models for chemotaxis, the spiral model, the Barkai model, and information about the chemotaxis network. Then they have this model paper, ethanol and acetaldehyde metabolism in human beings. This is kind of a paper you can go through. Another model paper, SMAD, nuclear cytoplasmic shuttling in a mutated system. So this kind of goes through that paper, and then you have this catalog of Jupyter notebooks, and that's the whole course. So this is a nice course to go through if you're interested in multi-scale modeling, kind of using a lot of different physics tools and other types of tools to do modeling in. So any comments or questions about that?
Just to say that that was from that course website was from a postdoc in James Glazer's lab.
Okay, yeah.
Yeah.
Do they host this?
Oh, sorry. Is it a course that's available? Is it available online?
Well, it seems like it's the course material that they're putting together. Anyway, that was all. It was like he mentioned this at the beginning of one of their what's he doing? He does this graduate course like network dynamics or something like that. It's basically going through so how do you pronounce it? Soros system biology text. Yeah, I'll try and find more info.
He has something on YouTube. Gleisner has, a YouTube channel.
Yeah.
As they do these days.
Yeah, that's I don't know. Like, I know that's you mentioned it's from James Glazer's group, so yeah, it's kind of that take on it. Very much biophysics, and also a lot of there's some dynamical systems in there. There's some kinetics and other types of systems biology analyses and so forth. It's really it'd be really fun to go through to see.
Yeah, for sure. For sure. This is sort of on top. It's been doing a lot of like physics-based modeling. We're using that for in this case for ultrasound. But really interested in because it's so computationally demanding, really interested in following up on some of these physics and informed neural networks. And anyway, I this weekend I put together a couple pages of kind of notes and references, and I'm trying to. I'm sorry, I'm not at my computer, but like multiscale behavior is one of those things that the Caltech professor. I'm blanking on her name. It's like anemia. Again, I'll find links. But if anybody's interested, yeah, I'm definitely looking for some help, and these are interesting ways to solve finite element problems. We're currently using these finite difference time domain models, but it's really, yeah, really interesting for modeling multiscale behavior too.
Right. And what are the connections there? Like,
In terms of the connections to the embryo.
yeah, I mean, as an example, like what's the connection between finite element and multi-scale models? I mean,
Well, just so finite elements, you might well, the multiscale, the models get quite complex, right? Because you've got to have kind of like a mechanistic model at every level, and the real benefit with these kind of neural operators is that you're turning it into a machine learning problem, that you're solving with this kind of automatic differentiation that is actually just learning the mapping between domains, and there's some real efficiencies, and in terms of doing this, computational efficiencies. But you preserve the kind of multi-scale behavior. Whether it's necessarily as interpretable, or yeah, I'm interested to see what the differences are. That as we just, I'm still focused on implementing the boundary element and finite element skills to kind of complete out the normal kind of physics models. Sorry. Yeah, dogs and kids have a rush.
yeah.
My finite element analysis experience, just putting the trijunction into my cells, means that, maybe you see me better here, is a problem. Like, I'm kind of at the edge of it, saying that I have to refine my mesh, because I've got things that are too disparate in size. Like, the side of the cell is too large compared to the trijunction.
Okay.
I've had some trouble in that with that size difference. Even though the finite element analysis, you're supposed to be able to do chemicals, or look at the physics of, say proteins. That's too big a difference in size to translate that back to the cells that I'm drawing. Anyway, that's my experience with it, is trying to do the multi-size, multi-layered model, at least in COMSOL, doesn't seem to want to work.
Yeah. All right, so that's good. Yeah, thanks for that discussion. That was good, especially putting the last thing that we were talking about in context and how that's useful. So let me move on to some papers. Okay, so let's see. me talk about this one. So this is a paper from Biological Theory. This is in a special issue called Darcy Thompson's Conceptual Legacy. So Darcy Thompson, of course, was alive, over 100 years ago. He wrote a book called On Growth and Form, and he proposed that a lot of growth processes and a lot of these anatomical structures in seashells and in other types of systems they had this mathematical aspect to them. So there were mathematical scalings, and you could use the golden mean to say describe a seashell and things like that. So he was very much mathematically oriented, and he wasn't necessarily a Darwinian, but he did kind of bring these ideas up in sort of put a mathematical cast on them. So that's interesting, and people have tried to follow up on his work. There's been a lot of stuff in terms of simulation, in terms of the mathematics, and there's been some interesting work on this. So this thematic issue is kind of revisiting this conceptual legacy. So this is a paper called Biological Theories of Morphogenesis based on holistic biophysical thinking. And this is from Amy Shire and Alan Rodriguez, who I can't remember. They work at a university in New York City. Not sure if it's Columbia or one of the other universities there, but they do have done a lot of really interesting stuff with respect to morphogenesis. So this paper is relatively new. It was, I think, accepted last month. So let's read through the abstract here. So the abstract reads, the roles played by physics in the study of the life sciences have taken on many forms over the past 100 years. So this is, again, this sort of physical, mathematical approach to, I guess, life sciences in general, but developmental biology and, biology more generally, or more specifically. Here we analyze how physics can be brought to bear on the contemporary study of morphogenesis, where new tissue scale forms arise of simpler, more homogeneous initial structures. We characterize how morphogenesis has been studied through reductionist approaches and discuss their limitations. We suggest an alternative way of approaching morphogenesis that begins with a consideration of the whole, which may also serve as a fruitful mode of scientific inquiry. So what they're proposing is that, that there's been a lot of the work on morphogenesis has been this sort of reductionist approach where you're looking at specific genes and manipulating them and seeing what their effect is on the phenotype, or you're looking at specific manipulations and looking for mechanisms, which are usually, molecular, but you can also see them at the morphological level. And so, if you look at the reaction diffusion model, that's, I guess, reductionist because you're looking at different morphogens and sort of their behavior in specific places in the embryo. So basically, though, I think a lot of the experimental work is what they're critiquing. So they're proposing this alternative way of doing this by considering the whole. So through historical analysis of concepts relevant for contemporary systems biology, we illustrate how physical thinking oriented towards the biological whole, or holistic biophysical thinking, was exemplified in the biological theories presented earlier by Darcy Thompson and Per Albrecht. So Per Albrecht actually was another person who's done a lot of things. It did things with allometric scaling, looking at that, published a number of works with, like Stephen Jay Gould and other people. So it's interesting. He had an interesting career as well. In any case, they're drawing back to those works, and they're trying to bring that work forward. We contrast the use of physics to develop the conceptual foundations of holistically oriented systems biology with the more prevalent contemporary use of physics that focuses on technological development and quantification embedded with reductionist strategies. So they're kind of saying, you might use something like next-gen sequencing or some of these imaging tools where you're looking for, patterns in the whole embryo, but you're kind of looking at features. You're looking at, they want to kind of bring these theories to the kind of techniques that we use today to look at morphogenesis. So that's kind of where they're going with this. An in-depth contemporary case study. As an in-depth contemporary case study, we describe how in our research we have used holistic biophysical thinking to develop biological theories of vertebrate morphogenesis that account for and extend beyond genetic, molecular, and cellular processes. We propose a theory of skin development where both molecular and morphological patterns were orchestrated by emergent physical processes at the supracellular scale. So this is this work they do on skin development where they have like a model of the cell, like a layer of epithelial cells, but also have this genetic circuit that fits into the model. And so that's what they're going to talk about in this paper. But basically, there's this emergent physical process that happens at the layer of cells at the tissue scale, and that's kind of what they focus on. We further posit that holistic biophysical thinking at the supracellular scale may advance theories of diseases such as cancer. So that's the obligatory call for applications. In any case, they talk about holism and they talk about reductionism. And so, the idea is that, of course, uh, biological systems or whole takes precedence, holism, and where physics, or what we will refer to as biophysical thinking, is central to the task. To conduct an analysis on what we call holistic biophysical thinking, we'll trace two biological theorists who are pioneers in the use of observation combined with physical thinking to characterize the dynamic structures leading to the creation of animal forms. So this is where they talk about Darcy Thompson and Per Ulberg and kind of going through that work. So first we discussed the two forms of reductionism, time and scale. So this is where we have reductionism in terms of time and reductionism in terms of scale. So this is an interesting way to divide this up. So of course we're familiar with reductionism with respect to scale, which is basically spatial reductionism, or like, reducing things from cells to genes to, who knows what. But, it's basically going from the whole down to the mechanism. And so we're familiar with that. And it's usually spatial in an embryo because we want to know, like maybe in a specific cell, what the mechanism is, or in a, maybe in a bunch of cells, what the specific mechanism is. So there is a spatial component as well. But there's this idea of reductionism as sort of reductionism in time. And so that means that, you're trying to narrow down the window of some change. So we might want to know, say, for example, we see a change in the embryo, some stage of development. We want to kind of reduce that down to the exact precise mechanism and time that happens. So there's some mechanism that we want to know that kicks everything off, and that's what we want to know. And so, that doesn't necessarily leave room for things that are sort of these systems that have feedback, a perpetual feedback, or systems that sort of are self-propagating. Those are kind of left out of that kind of analysis. So, we've covered papers where they talk about these kinds of things in more detail. So this is just one take on this. I think it's an interesting take, but just be aware that this isn't like kind of a novel thing, but it's interesting the way they're doing this. So we suggest that a reductionist approach precludes the possibility of accounting for properties that are created across time, or properties that must be elaborated temporally or across scale, properties that emerge at a scale or level of organization larger than the constituent parts. In contrast to reductionist approaches, we then articulate an alternative investigative approach that we call holistic. So again, this is where, it's greater than the parts. we provide our view of its key elements. Number one is the importance paid to direct observation of a biological system. So this is where we have holism, where you can observe the biological system as a whole and see what that looks like. Number two is a focus on processes and relationships among the parts of the system, which can lead to awareness of emergent properties of the whole. So the first key element is observing sort of the overarching scale of something that's emergent. So for example, I can look at an embryo and I can see like movements in the embryo. I can see changes in the embryo that are sort of where the emergent properties of some some developmental process occur. And I can observe that. I can't really observe any of the mechanisms or processes or relationships. I just observe it from sort of the sort of the 10,000 meter view. So the second part, though, is to kind of focus on the processes and relationships amongst the parts of the system. So when you see this phenomena happening, you want to know what parts are involved, how they're involved. And so you can't really say something is emergent unless you're aware of its emergence, and that's kind of the point. Emergent properties are those that cannot be characterized or predicted solely by an understanding of the intrinsic features of the parts of the system. So we know that emergence is sort of the greater than the sum of the parts. So if we were to take like a bunch of reductionist studies looking at the action of specific genes or the action in specific cells, we can't simply put them end to end and get a complete description of the emergent phenomena. There's something else there that we need to describe. So this is kind of the problem that we've had with, a lot of things in biological modeling, but also a lot of things in understanding, how, say, we can move from the scale of individual cells to the scale of, say, tissues or embryos. Okay, so this is the standard definition of emergence, putting it in the context of developmental biology. Then they talk about Darcy Thompson, who pioneered a holistic approach that applied biophysical thinking to the consideration of systems level morphological properties. So he adopted this as the approach where he took, whole organisms or, things like that, and he did a mathematical analysis and described sort of the emergent properties of it using mathematical properties. Didn't really think too much about what was going on underneath that whole, the entire emergent process, but gave a description of the emergent process itself. Thompson adopted this approach because for him, the whole is not merely the sum of the parts, but rather the result of relationships amongst them. So, again, you have, say, like a seashell or like a fish that was, growing, and you can describe that with mathematical laws, and then you can maybe make some statement about the underlying biophysics. Thompson's work was predicated on the use of observation, reminiscent of how natural historians look at specimens as the basis for generating physical insights and inferring mechanisms. And so Thompson's holistic biophysical thinking can be brought to bear on the study of many processes in development as well as disease. However, to concretely contrast holistic biophysical thinking and reductionist approaches, we focus here on one particular biological context, and this is where we get vertebrate organogenesis. And this is the onset of vertebrate organogenesis specifically. So for that sort of model system, we can turn to the work of Per Albrecht, who we suggest can be seen to exemplify holistic biophysical thinking. In contrast to perspectives that focus on the instructive roles played by chemicals and molecules on patterning, which include the Turing reaction diffusion model, as well as some other models. So in contrast to perspectives that focus on the instructive roles played by chemicals and molecules on patterning, which includes Turing morphogenesis and some other related approaches, Albrecht posited that the physical processes amidst collectives of cells play a fundamental role in generating form during organogenesis. So basically, you're looking at the physical processes like we talk about with respect to tensegrity. You have these physical processes that play a role in collectives of cells and shaping those collectives of cells, and then this contributes to organogenesis. And the idea is that has emergent properties, which we can then describe, by looking at lower and lower scales. But the level of analysis that's most interesting is at the biophysical level. Well, why don't we go? Yeah, why don't we go to this part here where they talk about temporal or length scale reductionism. So the process by which living systems create their forms, or morphogenesis, are commonly acknowledged to be nonlinear and complex. They are nonlinear because, for instance, a twofold change in one protein in a pathway does not lead to a twofold change in the final output of the pathway. So you can have all sorts of fluxes and things within different pathways, and it can lead to some final output that is either greater than the sum of its parts or less than the sum of its parts. They're complex because the components within the system are interrelated such that changing one variable leads to the automatic changes in other variables. So this is where you have, all these different variables that, lead to, if you modify one thing, a bunch of other things get modified. And this makes it difficult to isolate the effect of varying one component while holding all other components fixed. So typically in an experimental setting where we want to sort of make a manipulation and we want to test this with a control, we usually fix everything except for one intervention, and we make the intervention and we observe the output, and we can say that the thing that we intervened with, the thing that we manipulated, is the thing that caused it, or at least was responsible for that. And then this is a path towards determining causality. So it's very important that we get that kind of, we get those kind of things, we control everything else in the system and we focus only on the thing we're interested in. Of course, the problem is in morphogenesis is that there are a lot of things that are changing all at the same time, and it's very hard to fix those things, and make them sort of constant, and then do some targeted intervention. So that's why we have morphogenesis being both nonlinear and complex, and why it's very hard to use sort of a reductionist technique to get at these problems. When faced with a poorly understood complex living system, an investigator must adopt a research strategy in order to further their understanding of the system. While they are often only implicit, research strategies used to investigate the emergence of living form have become dominated by reductionism. So these are some references that talk about the different types of reductionism. So of course there's temporal reductionism, which we talked about, which is of course, where we think about focusing on finding an initial state or source that holds the information necessary to provide instructions to generate the final form that is an interest to an investigator. So basically having this sort of initial state that will then give you the output that you want. And so like I just said, sometimes processes are nonlinear with respect to output. And so, having an initial state isn't always going to give you that final output or that final form. So, so for example, this type of reductionist notion is present in the idea that the genome stores information sufficient for specifying the skeletal pattern we see in our hands and feet. So, reading things out from the genome is one example of this, where we don't really have a set of blueprints in the genome. We have genes that are expressed, and those genes can be expressed in different ways. Furthermore, you can interfere with that to modify your hands and feet so that you may have fewer or more fingers than you should. And these are complex processes of gene expression and gene regulation. But of course, using this reductionist approach, we can't necessarily get at this. We still have to think about it in terms of, reading out the genome and reading out the instructions. In such a view, individual cell biology, as well as collective cell processes that occur as a tissue, takes on its structure. are seen in processes that merely execute the information stored in the initial state. So, thinking about biology as an executable, and this is actually relevant to some of the things we've been talking about with synthetic biology, because, we talk about executing things from the genome. There's been a lot of work in bacteria where you're executing sort of programs in the genome, you're reading out things. And for simple, if you're, trying to express simple compounds, maybe that's a good way to do things. Maybe it's easy to sort of execute the genome and get like some compound or some, enzyme that you want to produce. And that's fine. But like when you're doing something in the realm of morphogenesis, maybe this is a little bit harder because you're not just, uh, building chemicals in the system, but you're also regulating their concentration, and you're regulating this over space, and many cells are doing this collectively. So it's hard to get this sort of executable model to work in pattern, to explain pattern formation. Concentrating efforts on identifying regulatory elements within the genome and the supposition that their identification will reveal such instructions as determinative is therefore an instance of temporal reductionism. So, they kind of go through temporal reductionism in this section, and they give some more examples. And then they talk about length scale reductionism, which is where you have this more reductionism with respect to scale. So this is where we go from, like the genomic scale to the morphological scale, and there are different ways you can think about this. So from molecules to cells to tissues to organs to organismal. This is called the hierarchy Schnell of scales, which I guess is from Schnell 2007, and they talked about these different scales and their properties. So there are a lot of ways you could, I guess, parse the hierarchy of biology, but Schnell provides this scalar approach, or this scale dependent approach that might be interesting. Much of experimental developmental biology today has been influenced by the seminal genetic screen in Drosophila, identifying key signaling genes and transcription factors involved in structural patterning. Today, 40 years worth of genetic and subcellular scale focus has led to a meticulous catalog of key genes, as well as a corresponding non-coding gene regulatory regions, biochemical signaling pathways, and cytoskeletal machinery. So we basically, in a lot of ways, we think of events at subcellular scales as being considered to be fully responsible for mechanisms generating structure at larger scales. And this is from Nyhout in 1990. For example, master gene expression patterns or morphogen gradients were often viewed as responsible for setting the pattern for structure seen at larger scales. However, the possibility that morphological features emerge at the tissue scale that are not reducible to smaller scales has received far less attention. So they talk about this, the sort of struggle between thinking about, things that are happening at the molecular scale, translating to the morphological scale, and whether they're, sort of one thing is responsible for the other, or merely if they're sort of influencing the other. And so, sometimes you have things that happen at the molecular scale that merely influence what happens at the tissue scale. And there are other things that we're not really describing that give maybe a more full description of what's going on. So they kind of go through this. This is a nice paper because they kind of go through this history, and they talk about the contributions of Albrecht and Thomson and bridging that to the modern era, and then talking about epigenetics and kind of thinking about this, kind of giving this new way of thinking about it, or at least proposing a way, at least to experimentalists, so that they can think about emergence and they can think about their model systems, but also in this context of this multi-scale set of relationships. So is this something that is just merely repeating what we've been talking about with respect to multi-scale models? Or does this have something to contribute to this area? I think it's the latter. I think, they're kind of coming up with a number of really good points here that kind of fit into this multi-scale modeling effort, because we're thinking mainly about how to structure the computations and structure the problem, but this is really kind of linking this to actual systems and developmental biology. So that's the paper. Any comments or questions about that? Okay. Well, if there are no comments or questions, I guess that's it for this week. Anything else we want to talk about before we go? Or?
This tensegrity, the tensegrity that changes shape. Like you push it and it completely changes shape. Is there another name for it, or keyword? Well, keywords, so I can look it up. I don't know.
Metastable.
Metastable? Oh, okay. That might work.
You didn't say perfect.
Anyway, I think I should mention it in my thesis.
All right. That sounds good. All right. Thanks for attending, and talk to you next week. Bye.
Thank you.
Thank you. All right, now I'd like to talk about a few papers in tissue biology dynamics. So the first one is about dynamics from a single snapshot, and the other paper is a review that kind of calls into question the way we study tissue biology, and in fact connects to the paper on sort of emergent properties and holism that we talked about earlier in the meeting. So the first paper is this bioRxiv preprint on temporal tissue dynamics from a single snapshot. So this is Jonathan Sommer, Shia Manor, and Uri Alan. And so it's a systems biology approach to this problem. So the abstract reads, physiological and pathological processes, such as inflammation or cancer, emerge from the interactions between cells over time. However, methods to follow cell populations over time within the native context of human tissue are lacking, since tissue biopsy offers only a single snapshot. So, we have these static snapshots of tissues, usually tissue biopsies, and we can look at them under a microscope. They don't tell us a lot about how a process like inflammation works, or how cancer sort of metastasizes, or even how wound healing proceeds. Although we do have some good experimental systems of wound healing, usually those aren't sort of the standard for kind of looking at these things. Here we present one-shot tissue dynamics reconstruction, or OSDR, an approach to estimate a dynamical model of cell populations based on single tissue samples. So this is again using a dynamical model and modeling these cell populations, and is using a single tissue sample as input. OSDR uses spatial proteomics data to learn how the composition of cellular neighborhoods influences division rate, providing a dynamical model of cell population change over time. We apply OSDR to human breast cancer data and reconstruct two fixed points of fibroblasts and macrophage interactions. These fixed points correspond to hot and cold fibrosis, in agreement with co-culture experiments that measure dynamics directly. We then used OSDR to discover a pulse-generating excitable circuit of T and B cells in the tumor microenvironments. These are two types of cells in the tumor microenvironment, and that there's an excitable circuit that generates a pulse, suggesting temporal flares of adaptive anti-cancer responses. OSDR can be applied to a wide range of spatial transcriptomic or proteomic assays to enable analysis of tissue dynamics based on patient biopsies. So patient biopsies are very easy to access, and you would like to model things sort of not as a theoretical thing, but things that might happen in a single patient or a process that is sort of otherwise hard to image. So this is where we can model different aspects of the dynamics of these tissues from the single input, and it gives us this, pretty reliable set of outcomes, including this sort of excitable circuit, which models these temporal pulses. So again, this is an overview of what they're doing, this one-shot tissue dynamics reconstruction. You have a biopsy of some piece of tissue. You get the spatial proteomic snapshot, which is basically like imaging the protein distributions within the tissue. Then you get this dynamical model estimation from the single static model. And then you have the cell population dynamics that you can model from the differential equation, or from the you have the dynamical model estimation, which is a function, and then you can extract the cell population dynamics from the phase portrait shown in 3. The second part is neighborhood composition maps to a location in state space. So you look for neighborhoods, which are these local groups of cells, usually within a certain number of millimeters, and so of a radius. And so you can actually model each neighborhood and its composition. And this maps to a location in state space. We see up here the phase portrait number 3 in A. You have this, you can also map single cells to this phase portrait and get a state space estimation. You also have cell division and death markers determining change in tissue composition. So again, you have this neighborhood, you have different activities in this neighborhood. So for example, you might have division events in the neighborhood, and you might model those as cells that are, or cell populations that are moving around in state space. So this neighborhood evolves in state space. And so this all lends itself to control theory, because we can see we have x dot as the dynamical estimator, and we have the state space. And so we can actually do some state space-based control theory. And so this is where this is going. We also have breast cancer tissue with neighborhood mapped to a location in the phase portrait, estimated by OSDR. So again, we have the static snapshot, the spatial distribution of cells, of fibroblasts and macrophages. So it's a mixed population. You have these neighborhoods that are locally salient spatially, and then those map to this phase portrait where you have fibroblast density versus macrophage density. So it's a little bit different than the number of cells of different types in the first examples. You now have this density phase portrait, and you can map the spatial distribution of cells to this. Finally, we have probabilistic division and death events for each cell. And if we do this, we can look at the stochastic trajectory of tissue composition over time. So basically, we want to know how many macrophages and how many fibroblasts are present at any given time. And we can model that by looking at the birth and death dynamics of each cell population type. So we know what the starting number of cells is. We look for doubling events. We also look for apoptotic events, necrotic events, but also cell divisions. And then we put all that into a trajectory which has these stochastic dynamics. And we can look at the proportion of macrophages and fibroblasts in the tissue. Some of the other things we can look at division events and their thresholds. So we can have markers such as Ki67, which is a marker of cell division. We have this amount of Ki67 in the cell over time, and we can look at like the amount of this over time relative to a certain phase in cell cycle. So the G2M transition in cell cycle, which is between 24 and 32 hours, we can see that the Ki67 concentration rises above threshold during that period. And so we can say that when we have those dynamics, that we will have a division event, so we can actually calibrate our division events to actual events in the molecular milieu. And we can link that to cell cycle, and we can link that to these population size-based models, because we can generalize this across many cells, or we can alternatively model Ki-67 as a stochastic process. So Ki-67 concentrations might shift over time depending on where the cell is in the phase space. So we can do all this, this is nice modeling work. You can also predict a cell's probability for division using the number of cells of each type within radius r. So in other words, if we have a lot of cells of a certain type within radius r, we can predict the cell's probability for division. So is it being crowded out? Is it something that is are there cells nearby that are inhibiting its division? Or if there are no cells nearby, is that facilitating its division? And so we can actually look at these neighborhoods and we can get counts and we can predict the probability of division in that way. This whole technique, OSDR, is based on a probabilistic rate equation. So this is where we have a differential equation that has a couple of parts. So the first one are deterministic rate equations. So this is a differential equation which has divisions minus deaths. So we have divisions, which are births, minus deaths, and the balance of that is the net growth of the population, or the deterministic rate at which the population expands. The second differential equation is a stochastic equation, which is where we have this change in time. We have the number of division and death probabilities within a tissue. So we basically sum these up for the entire tissue. So we take each neighborhood, we take the probability of division and death, and we then sum that and get some sort of amount of net growth over time, or net reduction over time. A deterministic ordinary differential equation can be derived from the stochastic models using the expected direction of change. So this is with respect to the phase portrait. So we can actually go from the static image to the differential equations, and these are ordinary differential equations, and we can take that information and determine a direction of change within the phase portrait. So again, our phase portrait example here is fibroblast density versus macrophage density. And we know that like when fibroblast density is at a certain density, macrophage density is at a certain density, there's going to be a certain expected behavior. So we kind of figure out where that is in the phase space, and from that we can determine this direction of change, which is basically the slope of the change between two time points. And then they say that their method OSDR accurately reconstructs dynamics from simulations of known dynamical circuits. So you can look at different circuits. So for example, if we have one stable point in a population of two cell types, A and B, and there's a density of each, we can know kind of what this one stable point phase portrait looks like. We can also infer phase portraits from that. So we can actually measure it directly, or we can infer it, and they actually match up quite well. We can look at this for two stable points as well, where you have multiple stable points. You can do this for three stable points and for four stable points. And of course, some of these points are not stable. So you can look at unstable and semi-stable states as well. And it seems to work pretty well. It seems to, kind of replicate the dynamics. And so with respect to the hot fibrosis and cold fibrosis, they refer to these in vitro experiments which established hot fibrosis and cold fibrosis fixed points. So they're looking at fibroblasts in a cell culture. So there's a number of fibroblasts in a cell culture in a well, and then you have macrophages, which are cells in a well. So you have this macrophages versus fibroblast comparison. So when the number of macrophages is low, you have what they call cold fibrosis, which is where it can be from very few fibroblasts to very many fibroblasts, and you can have this mode of cold fibrosis. And then this mode of hot fibrosis, where there are a lot of fibroblasts, but also a lot of macrophages. And so this is where you're getting this growth in basically in fibroblasts, going from very few to very many fibroblasts without macrophages. So it's basically fibroblasts dividing without very much death or consumption. In the case of when there are a lot of macrophages, you have a lot of fibroblasts being produced, but a lot of fibroblasts being consumed or dying off. So the rates are, well, it's rate independent. It's really about like how many cells are dying versus how many cells are just dividing. You can have a lot of division with a lot of death versus just a lot of division. And that's kind of the point, that you have these fixed points that are stable. And, it just depends on different aspects of macrophage growth as well as fibroblast growth and the conditions under which they find themselves, which I think is kind of summarized in this point here, where fibroblasts and macrophages communicate via growth factors. So the growth factors also play a role in this. And so we see some more phase portraits of fibroblast and macrophage dynamics. And we see these phase portraits. So we see actually in D&E, we see this Ki67 above threshold for fibroblasts and for macrophage. So again, this is where you get a division event where Ki67, if you're above threshold for Ki67, you get a division event. So you can see that when you have this fraction of Ki67 above threshold, there's a change in terms of how many what the fibroblast density is and the macrophage density. So you can see that this fits into these different modes of hot and cold fibrosis. And so you can replicate this for T cells and B cells. They interact in a cancer microenvironment. And you get these phase portraits for cancer microenvironments as well, where you're looking at this Ki67, where if it's above threshold, you get division. If it's below threshold, you don't get division, and so you see this for T cells versus B cells. Okay, that was a nice paper to show that like you can take a single input and predict outputs. And in the first paper that we reviewed today, they talked about nonlinear outputs. So, for example, if you have an input to a process, that input is not, the components leading to the output are not always stable. And sometimes, in some cases, like in morphogenesis, it's kind of hard to predict an output given the input. They were talking more about like if you're reading out information from the genome, how that's not a linear process. How you might have something that, like a gene for something or something encoded in the gene, and it either gets degraded as it gets expressed and incorporated into a tissue morphology, or it interacts with so many things that it's barely recognizable. So there are these different aspects. But in the case of the last paper, we saw that you can use differential equations to predict some of these interactions and to understand that system a little bit better. So when we think about holism, we not only have to think about sort of this fundamental problem of interactivity, but also of the tools that we bring to bear. Sometimes we can bring to bear differential equations, and that's enough to describe some of these phenomena in terms of phase spaces, phase portraits. But in this case, this paper, they're talking about tissue biology in search of a new paradigm. So this is from Annual Reviews in Cell and Developmental Biology. And the abstract of this paper reads, animal tissues are made up of multiple cell types that are increasingly well characterized, yet our understanding of the core principles that govern tissue organization is still incomplete. This is in part because many observable tissue characteristics, such as cellular composition and spatial patterns, are emergent properties, and as such, they cannot be explained through the knowledge of individual cells alone. So again, we have to have that population perspective. We have to have that tissue level perspective to really understand the different aspects of, in this case, tissue organization, but its origins come from morphogenesis. Here we propose a complex systems theory perspective to address this fundamental gap in our understanding of tissue biology. We introduce the concept of cell categories, which is based on cell relations rather than cell identity. So this is interesting that they're doing a different way of kind of approaching this from a different way. They're not thinking about what the cell is or what it'll become. They're thinking about the interactions. And of course, that's kind of what we do in, one of the primary concerns of holism is this interactivity. So this is an interesting way to think about it. Based on these notions, we then discuss common principles of tissue modularity, introducing compositional, structure, and functional tissue modules. Cell diversity and cell relations provide a basis for a new perspective on the underlying principle of tissue organization in health and disease. So in this journal, they have a table of contents. So they talk about tissue organization through animal evolution, cell types and their evolution, and then cell categories based on cell relations. So they propose a number of different types of cell relations. They propose the primary supportive relation, the complementarity relation, the instructive relation, the supplier-consumer relation, a hierarchical relation, and the mutual exclusivity relation. So this is where we have these sort of trophic roles. Instead of thinking about what the cell is, if it's a fibroblast or a macrophage, we think about the relationship between them. And of course, not all fibroblasts will have the same relation to each other, nor to all macrophages, and vice versa. So we can take these relations and break it down. We think about those spatial maps that we saw in face portraits. We think about this across space. There's variation across space. There are these different relations maybe that are more relevant to density than to cell types, or more relevant to isolation than to cell types. So this is a way to think about this very differently than cell types. And of course, cell types might be conditional on cell type. So you might have complementarity relations that are different between fibroblasts and macrophages than you would find within and between fibroblasts. So it's an interesting way to kind of look at this. And so then they go on to talk about the modular organization of tissues, talking about minimal tissue units and higher order modules. So minimal tissue units are groups of tissues that are basically, like we talk about these sheets of epithelial cells or fibroblasts, and that's sort of like a basic level of minimal tissue unit. And then the higher order module is something where you get these compositional, structure, and functional tissue modules, basically where you get different types of cells in a tissue, different structures, and different functions. So this is an interesting figure. Two modes of cell type diversification and evolution. So this shows two different ways in which you get new cell types in evolution across species. And so the first example is induced constitutive transition, where you might get the origin of a new function. And this is where you get a new cell type where there's a set of environmental interactions. There might be an irreversible transition from one cell type to another, where there might be genetic assimilation, where you have a convergence of phenotype given different genetic backgrounds. Then you have trade-off resolution by division of labor. So in this case, you're also getting a new function. This is not induced but constitutive. So in the first example, we had ways of inducing different types of new cell types. In this other model, we have the cell type diversification, or the introduction of new cell types as the origin of a new function, and an increase in functional complexity. So we have sort of new cell types constructed to sort of form more diverse tissues. You might think of like macrophages as being part of that in relation to fibroblasts, where fibroblasts are kind of pushed. Sometimes you're trying to heal a wound, you want to produce a lot of fibroblasts, but you also want a lot of those fibroblasts to eventually, die off. And so you need macrophages to sort of take care of that. You can think of different ways in which that works, but the more functional complexity you have, the more cell types you need. Then you have this continuum of variation due to trade-offs. So this is where you have different functional trade-offs, and you have different numbers of cells or different cell types, depending on, what types of activities, functional activities, adaptive activities the organism is engaging in. And then, of course, you can have the segregation of gene expression programs, where you have specific genetic modules for specific cell types or to define different cell types. And this is based on an increase in morphological complexity. So first part is functional complexity, where you have a change in functional complexity or an increase in an organism. And then you have morphological complexity increase, where you get diversification of cell types in the morphology due to different types of gene expression programs that get specialized, or different trade-offs that need to be met in evolution. So these are the two different states here, or ways of thinking about tissue organization. So on the left, we have this cell type paradigm. So this is where everything is based on cell type. You have your different cell types. You have your epithelial cells, your fibroblasts, some other types of fibroblasts, and maybe some phages in there where they're all kind of in relation to one another. It may be in layers. And so they might be doing they might have a specialized role in this sort of ecosystem of tissue. But they're all it's all based on cell type. So, maybe there's some process that involves multiple cell types, and they have their own role to play in this. The second is cell relation paradigm. So this is where we have the same set of cells in the same organization, but we're interested in the relationships between, say, like fibroblasts and phages. So we can actually look at that relationship and say that's a relationship that counts. There's, in fact, maybe a different way to describe this relationship, where instead of, maybe cell type C and cell type D being in the same layer interdigitated with each other, they're actually paired off as different types of, cells that rely on each other. So you can think of this as relational instead of typological. And of course, you can bring things like relational biology to bear into this as well. So then that leads to these different types of relations that they propose, and of course you can map those onto this type of organizational scheme. Finally, we go over the cell relation table. So this is kind of laying out the different types of relational, this different sets of relations that they're proposing for for understanding cells and tissues. Instead of thinking about this typologically, we're thinking about this functionally in terms of relations. So for letter A, we have the primary supportive relation, where function A supports function B, or function B is supported by function A, and function B, of course, is the primary function. Function A is a supportive function. So one cell supports another cell. They both have to be there. The second is the complementarity relation, where each cell type contributes to a common function, and so they all both need to be in the tissue to produce that output function. C is the instructive relation, where you have an environmental or a sensor cell that's sensing the environment. It's sending a signal to an effector cell, and then that gives that effector cell the ability to differentiate into different types of cells. So there are these cell fate choices that are made that are contingent on a signal sent by a sensor cell sensing from the environment. Then there's the supplier-consumer relation, where you have a supplier cell. It's providing a growth factor to a consumer cell, or sometimes both cells are both supplier and consumer cells, so they're providing each other with growth factor. But in this case, they're both dependent on each. One is a clear dependency relationship, and the other is where there's a codependency relationship. And they call this asymmetric and symmetric. there's the hierarchical relation where you have, say, like a T cell that's reliant on a mast cell, and the mast cell is reliant on the adipocyte and the smooth muscle cell. And then, where I guess it would be the other way around, where the T cell or the mast cell is dependent on the T cell, the smooth muscle cell and adipocyte are dependent on the mast cell, the stromal cell, the capillary endothelium, the macrophage, and the epithelial cells are all dependent on the adipocytes and smooth muscle cells. So you have this hierarchical relation of, where you can actually take the cell or take the tissue and say that these cell types are dependent on others. And so you can actually think then about how this tissue might have evolved. What needed to be there first? What needed to be there after the first cells? And how did the tissue come to be? Finally, they talk about the mutual exclusivity function, or the mutual exclusivity relation, which is where you have cell type A or cell type B. So you have this sort, you can't have one without the other, or you can't have one in the context of the other. So I guess it's like you can either have cell type A or cell type B, but you can't have both in the tissue for various reasons. So if you look at these different relations, you see that they're kind of grouped into categories. Cell relation A and cell relation B have this functional dependency. Cell relation C has this information dependency. Cell relation D, cell relation E, and cell relation F have this existential dependency. And so it's interesting, how you have this sort of thinking in terms of primarily function, thinking in primarily in terms of information, or thinking primarily in terms of survival or conditional survival. So that's an interesting way to think about tissue, because it takes what we know about cell fates and expands this to, like, given certain cell fates, are other cell fates necessary, or other cell types necessary, and what is their role functionally and structurally. So that's all for this group of papers. Hope you learned something.